Hi, this is George with 573 Medicine, and in this video we're going to be going over connective tissue proper. So let's get started. As a brief overview, there are four types of tissue which make up the human body. The first is epithelium, the second is connective tissue, the third is muscle, and the fourth is nervous tissue. And we are going to be focusing on connective tissue in this video. So first, let's define what the extracellular matrix is. So here we have the basement membrane of the epithelial cell, and here we have the basement membrane of the endothelium of the capillary, which is a type of simple squamous epithelium. And between the basement membranes of these surfaces, as well as deep to the basement membrane on this side, is going to be the extracellular matrix. And this is going to be composed of things like collagen and glycoproteins, which are surrounded by this watery ground substance. And this functions in support of the cell and aids in the structural integrity of the cell. And it includes not only the basement membrane, but also this interstitial matrix, as well as cells in the interstitial matrix. So, for instance, fibroblasts, which secrete collagen. So connective tissue is derived from mesoderm, and it's found between the other types of tissue that make up the human body. The general structure of connective tissue includes collagen, elastin, and fibrillin. So when we think of collagen, we should think of compliance. And compliance is the ability of a vessel or a structure to distend upon changes in pressure. Elastin is going to be involved in elastance. And so this is going to be distensibility or elastic recoil of this structure. And fibrillin is going to be important in the connections between the connective tissue components. The next is going to be ground substance. So we just talked about our fibers. Now we're going to be talking about ground substance, which is the water, as well as cells. There are fibroblasts, which secrete collagen, as well as adipocytes, white blood cells, and mast cells that secrete histamine. And for the next slide, we're going to zoom in on fibroblasts. So if you're watching this as part of Dr. Duff's first year medical histology class at MU, you're not really going to be tested on this for your in-house exam, but this is going to come up so much later on in your career. And I think it's important to pay attention just this first time and be exposed to it. And questions in terms of general collagen synthesis are more likely to appear than the actual pathophysiology. But collagen synthesis is just something that is good to be exposed to right now. So collagen is initially synthesized as this pro-alpha chain, and this pro-alpha chain is going to be composed of glycine XY. And so another name for this is pre-pro collagen. And this X and Y with glycine is going to either be proline or lysine. So this is important. The most abundant amino acid in collagen is going to be glycine. And then it alternates between proline and lysine. The next step is going to be hydroxylation of proline and lysine, which requires the cofactor vitamin C. And so in somebody who has deficiency in vitamin C, such as scurvy, they are unable to get the collagen outside of the cell and into the extracellular space. And this should make sense that whenever somebody has scurvy, they have bleeding gums and mouth ulcers, and that's because there isn't that structural integrity of the epithelial cells, which are making up the mucosa or apical surface of the tissues of the mouth. The next is that the pro-alpha chain is going to be assembled into pro-collagen. And so if somebody has a problem getting their pro-alpha chains to assemble into pro-collagen, that's going to cause a disease called osteogenesis imperfecta. And people with osteogenesis imperfecta characteristically have easy fractures of their bones, and it's actually oftentimes confused with child abuse. And this should make sense because you don't have that connective tissue, that collagen, in the extracellular space to provide structural integrity of the extracellular matrix. After procollagen is made, this is going to be exocytosed into the extracellular space. And then an enzyme called procollagen peptidase is going to cleave the CNN terminals of procollagen and create tropocollagen. And someone with Ehlers-Danlos syndrome is going to end up having a deficiency in procollagen peptidase. And someone with Ehlers-Danlos syndrome is going to have distensibility of their joints. And this should make sense because the collagen fibers are just too long. There's abnormal extracellular matrix. The next and final step is going to be the cross-linking of tropocollagen via the enzyme lysyl oxidase. And this uses the ion copper as a cofactor. In someone with Menkes disease, they're unable to metabolize copper, and so this step is defective. And so people with Menkes disease have connective tissue problems, which would manifest in things like brittle hair. And this is because of the inability to have an appropriate extracellular matrix. And something else is that, remember, collagen is for compliance or distensibility, whereas elastin is for that elastic recoil. And elastin synthesis is really similar, but actually requires a scaffolding protein called fibrillin 1. So next, we're just going to kind of look at how collagen appears on H&E. So this is the skin. This is going to be the superficial, and this is going to be the deep part. So the epidermis is above the dermis, so the epidermis is superficial. And in the dermis, we have collagen. And this collagen is going to appear differently in the papillary dermis. So we can see these fibers as well as these nuclei, which are primarily going to be fibroblasts. And in the reticular dermis, the collagen is going to look differently as well. So next we are going to be discussing the morphology of connective tissue. So connective tissue proper is what we're going to focus on this video. We already have a video out on blood, and we're going to be discussing bone and cartilage in another lecture. So connective tissue proper can be defined as either loose or dense. So loose connective tissue can be areolar, adipose, or reticular, and dense can be regular, irregular, and elastic. So we're going to be zooming in on some structures. So this is the superficial part of the skin, and we are going to be looking at the dermis and the hypodermis. And this picture shows lymph nodes, and this is a important organ system which functions in immunity as well as clearing debris in the intercellular space. And we're going to be zooming in on one of the nodes that's part of this system, specifically the fibers inside the medulla. So this picture shows loose. So this slide is going to be dedicated to loose connective tissue proper. So this is going to be loose areolar connective tissue, and this appears like a meshwork of collagen and fibroblasts, and this is found in the skin, like the dermis, muscles, and capillaries. This is adipose, and this is found in the hypodermis as well as any other area where you'd have fat. And this is going to be oval and round globules. And this is going to be loose reticular connective tissue. And this is made up of type 3 collagen or reticulin. And this is found in the kidney, spleen, lymph nodes, and bone marrow, which should make sense because these are areas in which there is lots of extravasation and things like sinusoid capillaries, so blood cells have to get in and out. And so it would make sense that they're loose. And what it means by loose is that there's a lot of white or ground substance appearing in these types of connective tissue. And so I think it's best to appreciate that on the areolar connective tissue, because on the loose reticular connective tissue, you actually need a special stain to see that, so it's not as clear. Next, this slide is going to be dedicated to dense connective tissue proper. So dense connective tissue proper can be characterized as dense regular connective tissue. So it's dense because you don't see that white, you don't see that ground substance. The fibers are closely packed and run in one direction. So you can see these basophilic substances, which are going to be the nuclei of fibroblasts. There's also dense irregular connective tissue. So dense because there's not a lot of ground substance or white that you can see, but it's irregular because the fibers are running in all different directions. This is found in the dermis, sclera, digestive tract, fascia, and periosteum of the bone. And this is elastic connective tissue, which contains a lot of elastin and found in the arteries and lungs. And you actually need a special stain to see the elastic connective tissue. So that's it for this video. If you like the video, please like, comment, and subscribe, and feel free to email us anytime at realtalkmeded@gmail.com.